Firstly, thank you very much for inviting me. My name's Dominic Abrams. I'm a cardiologist. I work here in Boston, the United States, but I'm originally from London, as I'm sure most of you have already worked out. And I've had an interest in different forms of inherited heart conditions, one of which really sort of stemming from a lot of my training in London was what we would now consider arrhythmogenic cardiomyopathy. My background is in electrophysiology, so that's the way I sort of came to different forms of inherited arrhythmic conditions and cardiomyopathies. So there's always, from my perspective, a bit more of an arrhythmogenic focus on things. Other people come from a different perspective, maybe more training in dilated cardiomyopathy, so they may see that a little bit differently. But I think LMNA can be seen in two different ways. It can either be seen as an arrhythmogenic cardiomyopathy with left ventricular dilatation, or a dilated cardiomyopathy with arrhythmias, and ultimately we're talking about the same thing. So it just sort of depends on your perspective. But there's an increasing move now in the world of inherited cardiac disorders to really think of this on a gene-specific basis. So we would now think of this very much as LMNA cardiomyopathy. So this is a slide deck that I thought might be a good place to start. This is something that we go through with our families. Usually the first time we meet them. So this is a sort of an introduction to the condition and how we're thinking about it, and to a large degree, why we do what we do, and really sort of explain, well, why are we doing the ECG? Why are you doing an MRI? What are you looking for? What are the things that are important to see? So I thought this might be a nice place to start. Some people may be very, very familiar with this. Others this may be a little bit newer, but we'll see how it goes. So when we're talking about an arrhythmogenic cardiomyopathy, we think of probably the best definition, and there are more than one out there, is a cardiomyopathy where arrhythmia is an early or dominant feature. And I would say that is true for LMNA. And this, we think about different arrhythmias. This can be atrial arrhythmias such as atrial fibrillation, ventricular arrhythmias, and also cardiac conduction disease as well. So we all know that's a very important part of the overarching phenotype of LMNA. So because in medicine we like to use lots of different confusing terms for the same thing, just to sort of set what we mean by arrhythmogenic cardiomyopathy, this is sometimes the original description was something called arrhythmogenic right ventricular cardiomyopathy, which we now very much associate with a different gene called plakophilin-2 and a few others. But it was sort of increasingly recognized over time that there were patients with left dominant or left ventricular arrhythmogenic cardiomyopathies. So really using the term arrhythmogenic right ventricular cardiomyopathy made no sense whatsoever, because we're talking about a different ventricle. But just to understand where that term comes from and what that term means. But now, as I said, we very much think of this as a LMNA cardiomyopathy, and that's what most people would use in terminology to describe the different features we see associated with genetic variants in the gene LMNA. Next slide, please. So I don't know how well these ECGs are going to show, but just to go back to the history, the genetics was first defined in the Seidman lab. Cricket and John Seidman are two very well-known researchers in the world of cardiovascular genetics who I've had the privilege to work here with in Boston. And this was back in 1999 when Diane Fatkin and Callum McRae were the first authors on this study that identified patients with a combination of dilated cardiomyopathy and cardiac conduction disease, and found that LMNA, genetic changes in LMNA in a number of families, were the etiology behind this, and they did some very nice research that demonstrated that. So that was the first description in the cardiac world associating the gene LMNA with various forms of cardiomyopathy. Next slide, please. So, but we now know that sort of the spectrum of conditions, and certainly of LMNA, as we see in many different features or many of the different conditions that we look after, has kind of expanded. So we now think of ventricular arrhythmias, atrial arrhythmias, very important in LMNA. And so if you go back to that original paper from the Seidmans in 1999, many of the patients in those studies had atrial fibrillation and atrial arrhythmias as well, but now it's increasingly recognized the ventricular component is just as important as many people will already know. And so we sort of think of this very much as an arrhythmogenic cardiomyopathy with cardiac conduction disease as well. And that's also a part of the spectrum and all part of the features, of course, that we're looking for when we're investigating patients or families who we know or suspect may have LMNA cardiomyopathy. Next slide, please. So just to go back to a bit of basic anatomy and cardiac physiology. So obviously the 4 chambers in the heart, the right atrium collecting blood coming down from the head and the neck and up from the body. This is deoxygenated blood. Goes through the tricuspid valve, which we've drawn slightly open there, and into the right ventricle, and then is pumped up to the lungs, comes back into the left atrium, through the mitral valve into the left ventricle, and then is pumped around the body. And we measure cardiac activation electrically on the ECG, which shows us some important components. The P wave tells us that the atria have contracted and electrically activated and then contracted. The QRS complex represents the ventricles, and the T wave represents recovery. So although when we look at the ECG, really it's telling us a lot about electrical activation, it's also telling us a lot of different structural things that we see within the heart as well. Next slide, please. And that is sort of best represented on the 12-lead ECG, which you can see here. So what we showed you there obviously was just one single beat, but here we have a 12-lead ECG. And as you all know, the ECG electrodes or the stickers are placed on different positions around the chest, which gives us information about different parts of the heart, not just electrically, but also structurally as well. So that's sort of a really, really simple investigation that's been around for 100 years, but still really sort of the basis of what we do, and hugely helpful in understanding how the heart is functioning electrically, but also how it's behaving structurally. Next slide, please. So when we're thinking about the diagnosis of LMNA cardiomyopathy, what are the different things that we do? We'll go through these in a little bit more detail, but the ECG we've talked about, really important, really basic, and then cardiac imaging in different forms, both using the echocardiogram, very easy to do in clinic, and then also cardiac MRI. Those 2 different forms of cardiac imaging give us different but complementary information. The one main thing that we get from the MRI that we don't get from echo is evidence of myocardial fibrosis or delayed enhancement. On the echo, we get different things such as how well the heart's relaxing and all sorts of different pieces of information, but the 2 are very, very complementary, and we use both at different times during the investigative strategy. And then heart rhythm monitoring using Holter monitors or different patches that are now available, also very important. To look at both cardiac conduction at different times, what's happening when you're resting, what's happening when you're exercising, and then also any atrial or ventricular arrhythmias that we may see that may be not evident on a 12-lead ECG, and very important in terms of how we think about risk. And then genetics is very important as well. Obviously, we know the genetics is pretty well established in LMNA, the gene that encodes for the proteins. And so, obviously, once we've identified one member of the family who has a genetic variant in LMNA that we consider causal, and we can talk a little bit more about that, then obviously we can use that to cascade test other members of the families and see if they are at risk or if they're not at risk. Next slide, please. So just going through those different investigations, here you can see the 12-lead ECG we've already talked about. And then the Holter monitor, there used to be sort of 24, 48 hours total in terms when we had the old style ones, but now we have the patches that last for much longer, which obviously give you a lot more information. So those are really sort of important parts of the investigations we use from an ECG perspective. Next slide, please. And then also imaging with both echo and MRI, and we'll talk about those in a little bit more detail and what we're trying to identify. Next slide, please. So if we think about the cardiac cell, it may look something a bit like this, give or take some license from an artistic perspective, but we have lots of different components. We have at the top, we have the membrane of the cell. And in the membrane, in the red, the blue, and the orange, you can see the channels that conduct sodium, calcium, and potassium in and out of the cell, which cause the cell to electrically activate. Also in the cell membrane, we have the desmosome, which is involved in many other forms of arrhythmogenic cardiomyopathy, and that then binds through the intermediate filaments to the sarcomere, which you can see on the left looking like a spring-like structure, which is the actual contractile apparatus of the cell. And over on the far right, we've got the nucleus and the nuclear membrane, which surrounds the nucleus. And lamin encodes for two proteins, lamin A and lamin C. There's others as well, but we're really talking about A and C, and those are integral parts of the inner membrane of the nuclear lamina. And that's really key in terms of the function of the nucleus, and that's something that's sort of a fundamental role of what lamin does. So it supports the nucleus, it's involved in keeping the structure of the nuclear membrane intact, and also plays multiple different other roles as well. It can be involved in other gene regulation, so switching genes on and off. It can be involved in regulation of DNA and also transport of different things around the cell. So, as is often the case in probably all of these that we're showing here on the slide, they have one sort of major role, but also have multiple complementary roles as well that we're still beginning to work out. So that's where we think of lamin from a sort of a molecular biology perspective, where it's located in the cell and the role it plays. Next slide, please. So this is what the lamin gene and the protein look like. So you can see at the top is the gene, and what we've coded there are the different exons from 1 to 12. So the exons are the coding regions of the DNA, which are separated by the introns, and we'll go through that in a little bit more detail later in the slide. And the different shades of blue relate to different domains in the gene and then ultimately in the protein. So we have, you can see the head domain, the central rod in the middle, and then the tail domain at the end. This gene codes for both lamin A and lamin C. So what we call differential splicing or differential isoforms that come from the gene means that this one gene, LMNA, can code for both lamin A and lamin C. And as you can see there, lamin A is a little bit shorter. It's missing the terminal part of the tail domain compared to lamin C. But lamin is a gene, sort of probably the greatest example in the human body of a gene that explains or that displays pleiotropy, which means that it can cause lots of different things. So lots of different conditions can arise from the same gene. Obviously, cardiomyopathy is very well described. Different forms of skeletal myopathy, classically with muscular dystrophies, which have onset early in childhood, things such as Emery-Dreifuss, and then other rarer forms or rarer conditions such as lipodystrophy. Some families have a history of diabetes, and also the very rare Hutchinson progeria, which relates to very one specific change within the gene. So this single gene, and based on its very central role in the cell around the nucleus, can cause multiple different phenotypes that have all been associated with it. Next slide, please. So what we're looking for specifically when we're evaluating patients with a lamin phenotype, the first thing is the ECG. So we know that cardiac conduction is an important component of the overall lamin phenotype. So we're looking for any evidence of conduction slowing between the atrium and the ventricle through the atrioventricular node. And that's represented between an increased distance between the P wave at the beginning and the QRS complex, which represents ventricular conduction. So basically the electrical impulse is taking longer to get from the atrium through to the ventricle than it should do. We also know that there can be conduction changes within the ventricle. That's typically shown as left bundle branch block, where rather than being a very narrow QRS complex, it takes on this broader M shape. And then we often see various forms of T-wave inversion. Basically, that's telling us that the heart is contracting in an abnormal way, and so therefore would relax in an abnormal way as well. So T-wave inversion is a very important thing that we look for, and often can be an early finding when we're screening people we know who are at risk of lamin by virtue of family history, or because they have a genetic variant that's been identified in the family as well. And that can often be a sort of a subtle change that we see early on. Next slide, please. So that's what that can look like on a 12-lead ECG. You can see there's first-degree heart block, which we define as a prolonged PR interval of greater than 200 milliseconds. There's a classic left bundle branch block pattern, suggesting that conduction is slow into the left ventricle as well. And then T-wave inversion, you can see classically in V4, V5, and V6, where the T-wave is inverted, as opposed to being upright, which is the polarity or the shape that it should take. Next slide, please. And so this is just a description of, you may not see this very well, but this patient is, or this ECG shows atrial fibrillation and complete heart block. So you can see that we've got a left bundle morphology on the QRS complex, so the slow conduction through the ventricle. But the ventricle seems to be activating on its own. So there's no P wave because the atrium is fibrillating, and the heart rate, you can see the ventricular rate is very slow and very regular. So this is a sort of a classic LMNA presentation in a proband, often in their 30s or 40s. And when you do an echo scan, you'll see ventricular dysfunction as well. So this sort of demonstrates all the different findings that we can see associated with LMNA. But obviously what we want to do is identify people who are at risk of this before they get to this point and instigate different treatment strategies. Next slide, please. So when we're doing an echo, what are the things we're specifically looking for? So we can look for the size of the left ventricle. That's going to be very important. So we look at the dimensions of the ventricle when it's completely relaxed at end diastole. That's the relaxation. And we can assess the volume from that. And of course, we can compare that to the normal range that have been derived from many, many echoes over many years of doing this. We'll also look for any changes in wall motion and also the wall thickening. So we know in LMNA sometimes the wall can be thinner and areas of the wall may not move as well as other parts of the heart. So changes in the motion, changes in the thickening can also be very important. And we can assess the relaxation. So what does the diastolic function look like? How well does the heart relax? And then of course we're looking for how well the heart contracts. So that's the left ventricular systolic function or the ejection fraction, which is measured as a percentage. Normal percentage being somewhere between the low 50s and 70s, certainly in adults. And so we would obviously, that's something that we can measure very easily on each echo. Next slide, please. The MRI tells us a lot of those, a lot of that information. It's often slightly more accurate because we're sort of looking at the heart in 3 dimensions. And of course, the heart is a 3-dimensional structure. So it does tell us all these things as well. It tells us about left ventricular ejection fraction, wall motion. But as we said at the beginning, the thing it really identifies that the echo doesn't is areas of potentially inflammation or fibrosis, which, as you can see from the little heart image beneath the scanner, is that white streak in the muscle, which is shown largely as gray. So the gadolinium injection, that goes through the heart muscle and sticks to areas of fibrosis or scarring. Again, that's a very sort of important component of what we look at when we're thinking about the overall changes that we see in LMNA-affected hearts. And that's obviously not something that we can see on echo. Next slide, please. And then we've already talked a little bit about Holter monitoring. This is also really key. We're looking for evidence of single or isolated premature beats, which can either be atrial or ventricular. And if we go to the next slide, please, then we'll see that coming in a little run. Something we call non-sustained ventricular tachycardia. You can see there there's 8 or 9 beats together. And again, that's very important in risk stratification, identifying patients who may be susceptible to that continuing and going into an evidence of a sustained ventricular arrhythmia. So non-sustained is anything more than 3 beats at greater than 120 beats a minute. Sustained is greater than half a minute. So these are sort of very important findings that we can pick up, often asymptomatic, but are early clues that we can get that things, we need to change how we're thinking about that individual in terms of risk. And so super helpful information from our perspective and the patient's perspective as well. Next slide, please. And just to talk a little bit about family history and genetics, we sort of go through this because obviously we work very closely with the team at the Brigham, who I think you've already heard from Neil Lakdawala. And it's very important to look after the family as a family. So we think of the family very much as the unit of care. So we will see the children at Boston Children's Hospital, and the adult patients, the parents, will be seen at the Brigham. And we're very closely connected by a bridge, so there's a physical connection between the two. So it's about 200 meters from my clinic to Neil's, which is really helpful in looking after the family as a single unit. Next slide, please. So when we're thinking of genetics, we go through the patient's own genetic result with them. I think that's really important because obviously people want to understand what it means. So we go through this in quite some detail so they know exactly what all those different letters and codes and things mean. So this tells us a little bit about how our DNA is constructed. So it's a series of 4 letters, which are the nucleotides, C, A, G, and T. And they come together in groups of 3. And each 3 nucleotides will encode for a single amino acid, although there's multiple different ways that amino acid can be coded. and then also a stop codon, which is used at the end of the gene to stop the process of transcription. So amino acids are the building blocks that make up proteins. Lamin is a protein. And as we said, each gene consists of the coding regions, which are the exons, and the non-coding regions in between the exons, which are very much involved in regulation and other aspects of where and when genes are spliced or cut. So there's lots of, although we used to think that was all non-important information in between, there's actually a lot of important regulatory and functional information in the introns. And so because lamin has been very well studied over many years, we know a lot of the genetic changes, and those are familiar, which is very important in terms of how we interpret different variants or changes we may find. Next slide, please. So lamin is inherited as an autosomal dominant condition. We have two genes, two copies of every gene, one we get from our mother, one from our father, and the LMNA gene is no different. And so if there's a genetic change in one copy of the gene, that can predispose that individual to developing LMNA cardiomyopathy. So in this sort of very simple family construct, we've got a father who has the lamin variant, which is the altered copy of the gene, and one copy of the wild type, which is the unaltered copy or the normal sequence. The mother is obviously unaffected. So as you can see, there's a 50-50 chance that he will pass that on to any of his children. Here we've created a system so two children inherit it, which is statistically the most likely. But of course, it's a 50-50 chance any time that couple have a child, so it could be anything from 0 to 4 who are affected. Next slide, please. And sort of important concepts that we think about with any of the inherited cardiac conditions that we see are penetrance and expressivity. So penetrance we think of as a binary phenomenon. Does any individual who has a genetic change and therefore is predisposed to the condition go on to develop that condition at some stage in a way that we can identify? And that's a sort of a yes or a no. So typically we think of the penetrance in lamin as relatively high. So the majority of people who are genetically predisposed do seem to go on to get different forms of lamin cardiomyopathy, certainly. But that may be very different even within members of the same family, and that's what we term expressivity. So whether we think of that as mild, moderate, more severe, that's expressivity. So that's the sort of the variability that we see in all the individuals who have penetrant forms of lamin cardiomyopathy. So those are two things that we talk about that are important, just for people to understand what those could mean and what that could mean for them going forward. Next slide, please. And then we'll go through with families their specific genetics. So in this situation, we've got a premature stop codon. You can see at the top of the sort of different amino acids. There's just a series of 5, and then you can see at nucleotide position 1057. There's a change from a C to a T. And what that does, it changes glutamine to a premature stop codon. And this is about halfway through the gene. So this gene is going to stop what we call transcribing about halfway through. If we can go to the next slide, please, we'll see what that does. So here we have the 2 copies of the lamin gene in this individual. You can see that at the top, there's the whole gene. Copy of the gene that's the top codon, which is denoted by the asterisk. You can see that this is in the terminal portion of exon 6, and that means that the messenger RNA is not always transcribed incompletely and stops prematurely. And what we think happens is that this is recognized by the cell and then broken down by a process we call nonsense-mediated mRNA decay. And that's basically a defense mechanism that the cell uses to prevent an abnormal copy of lamin being produced. And so this is a mechanism we call haploinsufficiency, which is common in lamin cardiomyopathy, where that individual is essentially functioning with one copy of the lamin gene, creating lamin ANC rather than 2. So approximately that individual will have half the amount of the protein that an unaffected individual in that family would have. But we know that there's other changes in the genetics that can cause lamin as well. Go to the next slide, please. And this is a missense variant. So this is a single amino acid substitution caused by a single nucleotide substitution. Here you can see amino acid number 541, which correlates to the nucleotide position 1621. It changes from C to T. So the way we would write that is 1621 C to T. EGC encodes for cystic. Everything before that, all 540 before that and the 300 or so after that, will all be exactly as they should do, but there's just this one nucleus, this one amino acid that's different in the center part of the gene. And although that would seem a very minor change, these can have very significant effects on how the protein functions, on how it binds to other forms of lamin, how it configures within the cell membrane. So this is a very well recognized mechanism of genetic variation causing lamin cardiomyopathy. So we think of the two together. And then finally, in the next slide, please, what we would then do with the families is go through what do we know about this specific genetic change. So we'll say it's been seen before, it's been reported many times, it's either been shown to affect multiple family members, we call segregation, which is very important and for us to understand in terms of causality, or it was seen de novo, which means it's arisen in a patient for the first time. Again, that's important. And then we say, well, has this been seen in control databases? So we now have this huge resource called the Genome Aggregation Database, or GNOMAD, which is about 800 control subjects who gave their DNA very kindly for research. And it gives us an understanding of what genetic variation exists in the population. So this was only seen in one individual, which tells us that not everyone with this genetic change will go on to get the cardiomyopathy, but it's very, very rare in the population, suggesting that the majority do. So we sort of give a little bit of background about the specific genetics that are affecting their family, and then usually what we'll do is we'll sort of end with a plan for the family going forward and what that looks like over the next year or 2 years. So that's just a bit of an overview. That's what we use. Hope that's sort of given some interesting background. Many of you may be very familiar with all of it already, but I thought it was a nice place to start. We can get into a conversation and questions from here.